Ayla in 2016, and I have been mostly involved with a new project rather than plankton, but I'm still involved with plankton work. That's what I'm going to talk to you about today. So I've been based in the marine station in Villefranche-sur-Mer near Nice. And it's a place which is famous for plankton, and this since 1850s, when Haeckel came to Villefranche to do his first drawings. And then the marine station, which you see here, was created in 1880s. And the particularity of Villefranche is that we have a very deep bay here, and a canyon, and organisms that you usually find only far at sea, you can actually find in the bay at different times of the year. And I've put a little sample of these ctenophores, jellyfish, siphonophores, and all kinds of other organisms that you can collect very close to the laboratory. So that's why Villefranche is very well known for plankton. And my involvement with plankton really started towards the end of my career, but as a cell and molecular biologist, I had explored a few planktonic organisms. But I will start by showing you one of the Plankton Chronicles film, which gives you a little feeling for the plankton on the Riviera.
The sea here is an intense azure blue, reflecting deep underwater foam. A few miles offshore, the Ligurian current carries plankton, the multitude of organisms that drift with the current. Plankton includes microscopic creatures like bacteria, diatoms, and siphonophores, the longest animal in the world. Between these two extremes is a world of tiny algae, invertebrates with strange shapes and habits, and countless embryo and larvae. At certain times, under optimal conditions of light, temperature, and nutrients, some planktonic organisms, large and small, proliferate in spectacular bloom.
That's a night dive in the Bay of Villefranche. That's not kind of typical.
Over the past 200 years, artists and biologists, Perron and Lessoir, Bach, Barrois, Paul, Corotnet, and their descendants at the marine station of Villefranche-sur-Mer have drawn and studied the gelatinous creatures which abound in bays and along the seashore. Resembling miniature sailboats, velella are actually groups of individuals living together as one colony. They drift at the interface between water and air. At the end of spring, these cousins of jellyfish proliferate. Driven by winds and currents, they beach and form blue fringes along the coastline. Speaking of jellyfish, large groups of medusa often invade our coasts. For example, Pelagia noctiluca, which can sting and inflict nasty burns. but there are also countless microscopic jellyfish that pass unnoticed. Some of these micromedusa, like the fragile Clidia hemispherica, can be raised in the laboratory for scientific study. Among the planktonic animals is Cymbulia peroni, alias Venus slipper. Some spring day, you may find on the beach its slipper-shaped cartilaginous shell. Abundant in winter and spring, Cymbulia is a member of the large family of pteropods, marine snails that have adapted to planktonic life by transforming their feet into fins. Then, with the first hot days of summer, appears the elegant and transparent ctenophore, Cestus viridis, nicknamed Venus girdle. Finally, if you think urchins have nothing to do with plankton, think again. Like most animals inhabiting the seabed, before becoming adults, they live among the plankton as embryo and larvae. Here, they are easy prey for other marine organisms, forming an essential link in the food chain of the Mediterranean Sea. Only a few will survive, enough to perpetuate the species and fulfill their role in planktonic life.
So my interest in plankton actually came little by little because I was exploiting the plankton diversity to ask the right question to the right organism for many years as a cell and molecular biologist. This was a traditional strategy that has been superseded really by asking all questions to a few organisms. But I studied quite a few different organisms in plankton for different questions. For example, I studied sperm attraction in siphonophores because there the sperm is clearly attracted to the oocyte. And I studied polyspermy and axis formation and other aspects that I'll tell you about in ctenophores. And germ cell determination in chaetognat, because that's where originally germ cells were really discovered, and they're easy to study in these organisms. And in terms of berry, this is just to give you one example. This is what I studied, fertilization. It's so incredibly clear. You can see literally the sperm penetrate into the egg. It's a very large egg, about 1 millimeter in size. And also we noticed that there was a very startling phenomenon is that the female pronucleus, when it is emitted, is going to explore several sperm. It's typical case of polyspermy. And until it decides which sperm to fuse with. This is one of the sequence, for example, where the female pronucleus here labeled with a fluorescent dye is going to go toward one sperm for some reason which we haven't completely understood. It doesn't like it, it goes to the next one and so on. And this will actually be the next, the axis of the embryo. Here is a sequence where you can see that where the female polynucleus is emitted and then goes to a first sperm, rejects it, then goes to a second sperm, fuses with it, and this will be the main axis, oral aboral axis of the embryo. So anyway, these are the kind of questions I was interested in. And it's not until the end of my career that I became a kind of a generalist of plankton. And this came about because I have trouble really getting the next slide. Yeah. This came about because with a friend of mine called Eric Cassanti, we tried for several years to do an expedition which was more like a TV show about evolution than a real expedition. And it was a time when Darwin was being celebrated around 2000. There was going to be a 2009 celebration, but we did fail to do that project for many reasons. And we said, why don't we do a real expedition? And I had known Eric for quite a long time as a cell biologist. And we had even done a DVD called Exploring the Living Cell, where he participated. And I had also connections with a lot of the oceanographers. My best friend was Gabriel Gorski in the Marine Station. So in 2008, we started co-opting a number of people with different knowledge to do this multidisciplinary expedition. And we started looking for a sailboat because Eric was a sailor and didn't want to hear about any kind of boat besides a sailboat. And we were lucky enough to meet people who had a wonderful boat called Tara. They got it around the year 2000 from a great navigator who had won the Cup of America on the boat. He had been actually assassinated on the boat. And then the family of Agnes Bay, who is a clothes designer, had recuperated that boat and started with small expeditions. And the head of the company, the clothes design company, Etienne Bourgois, went along with our request for a boat for 3 years, which was demanding a lot. And we actually equipped the boat, and we left in 2009, and the expedition has been going on until today. Different expeditions have been taking place since that time, using basically the same protocols, of course, adding new experiments to the original experiments. But the idea was to explore plankton in all oceans using genomics and imaging. We had, with Eric, because we were cell and molecular biologists, we had good connections to the genomic people, and we were also very well-versed in imaging, so that we thought that was a real good time to explore something as complicated as the whole planktonic system using both genomics and imaging. This was very ambitious, but it's probably because we knew very little about oceanography, which in that case was a very good asset. So we convinced many people to join with no money. Actually, this was a very informal project. We didn't get funded because we tried to, but we were nobody in the field of oceanography or marine biology. And also because they said we don't fund collecting. And all we wanted to do was collect in the best possible way the whole ecosystem and analyze it and see what what came from these analyses. So anyway, everybody gathered the money that they had to be able to embark on the boat, because between the time we decided to do that expedition and the time we took off, only a year took place. So this was a fairly short time, but basically we accomplished what we wanted to do. People were very skeptical that with this fairly small sailboat, 35-meter long sailboat, one could do good sampling, but we had absolutely no problem, thanks in particular to the people in Villefranche, like Gabriel Gorski and some people from the Naples University and Roscoff Marine Station. And we had an excellent head engineer with us from Villefranche also. So anyway, altogether, we were about 200 participants for the first Tara Ocean, which lasted about 3 years, 10 core labs. Villefranche lab was one of them, and a very multidisciplinary approach, of course. And we had a few sponsors, the CNRS, of course, in France. The life science part was very enthusiastic, but the oceanography was extremely critical. And the European Molecular Biology Laboratory was also very enthusiastic. And of course, we had the chance to have the National Genomic Center, the French National Genomic Center, who was also very enthusiastic about being able to do some metagenomics. So we wanted to collect the same way at all stations, which we did. When you went on the boat as a scientist, it was not a question of doing your own lab work or project. It was to collect the best way possible, the same way your colleague had been doing it. We did that at 3 depths with very rigorous protocols. And we had a good instrument that we never lost, actually, thank God, because we only had one, which was the underwater vision profiler designed by the people at Villefranche-sur-Mer. And we collected, of course, the biological samples, trying to collect all the organisms from viruses all the way to jellyfish. And we, of course, nets and filters and a bunch of different ways to not miss anything. And we also collected about 25 physical parameters at the same time. So that was very special, because usually this is not done, and this turned out to be extremely useful, of course, to do correlation analysis. And of course, trying to put genomics and imaging together. This is very powerful, because if genomics and imaging tell you the same thing about these complex ecosystems, then you're sure you're not making mistake. If they don't, then you have to start wondering what are the biases. We made only a few observations. The boat was very small, but we had a few instruments that were very useful on the boat. But most samples we sent to the different laboratories, like Villefranche got the zooplankton, and Roscoff got the phytoplankton, and so on. The Genomic Center got RNA and DNA samples, and so on. We miniaturized a lot of these equipment, and as I said, we had no problem collecting in over 3 years, and it's still going on, and new devices have been developed, such as the ability to collect, for example, while the boat is going full speed. So this is actually the route that we followed for the initial Tara Ocean expedition. We had to reroute because there was this problem in Japan with the nuclear contamination. So we had to do a first round and then do the Arctic expedition later on. So plankton, I had been dealing with plankton actually since I was a young researcher. Here I'm in Friday Harbor with my son Noe, who turned out to be an extremely great collaborator with his partner Sharif. They live in Montreal, but I owe them a lot in the terms of being able to film and photograph plankton. Here we are, for example, in Japan, doing some photographing and fixing. Here we equipped one of the small cabin of the Tara with a imaging lab, and I spent a lot of time in there. So the Plankton Chronicles project emerged as a way to accompany the expedition and also make my colleagues understand what was behind the names. A lot of the geneticists and oceanographers didn't really know what these organisms were. They were very good at doing genomics or oceanographic things, but I realized that they really didn't know much about the complexity of this ecosystem. So that's why we embarked on these short films, the Plankton Chronicles series, just telling the essential about each type of organism. And then the books came later, first in French, and now it's in 5 languages. And of course, this was very important also for communication. And talking about communication, this is a little document we did with my son, Noe, and Chris Bowler, who was the head of that Tara Pacific expedition, just to show you what life is on board. Sorry.
When observed for the first time from outer space, Earth was called the Blue Planet. But we did not imagine that the ocean is inhabited by an invisible world, incredibly powerful, the marine microbiome. Millions of species of planktonic microbes that support life on Earth and contribute to regulating our climate. We humans depend on another microbiome, consisting of all the microbes in our body. They digest our food, boost our immunity, and help us fight viruses and bacteria. Without a healthy microbiome, we cannot survive. The marine microbiome is essential to maintain the balance of life in the ocean. For 3 billion years, phytoplanktonic organisms, a multitude of bacteria and protists, produced most of the oxygen on our planet. They continue to provide food and energy for the entire marine ecosystem. Plankton sequesters as much CO2 as all the plants on Earth, while producing half of the oxygen we breathe. The Tara Ocean Foundation launched the Microbiome Mission to study the marine microbiome, viruses, bacteria, protists, and larvae. The expedition will study the relationships between these microorganisms and the rest of the marine ecosystem. In partnership with laboratories around the world, the objective is to understand how the marine microbiome functions and how it adapts to stress, such as pollution and climate change. The ocean is the key to life on Earth. Follow the adventure and discover this invisible ocean life.
So, of course, you know what plankton is. and that it comes from the ancient Greek word for drifting. And that explains why you have organisms as big as jellyfish and as small as all these protists. And this, of course, is just a visible part, at least in a microscope. But we have to realize that there are incredible numbers of organisms that are even much smaller, like viruses and gyruses, the giant viruses, 1 to 10 billions per liter. So this turns out you cannot really analyze this and if you don't have genomics. And this was the main, of course, effort of the expedition. And the one that gave the first results, imaging came little by little, and I would say that by now the two are being used together very efficiently. So there was this first expedition. Eric Cassanti was the research director of that expedition from 2009 to 2012. And then came the one where we had to reroute because we couldn't do the Arctic first, and that was led by Chris Bowler in 2013. Tara Pacific is the one I showed you. I know this was mostly devoted to corals and a little bit of plankton, but mostly corals. Tara Microbiomes, the one I showed you the film about, was a 2-year expedition that you can see here that went all around the coast of South America and explored the Antarctic, which we had 1 or 2 points there before, and then came back by the coast of the coast of Africa. And there were also some expeditions that were devoted to microplastic. I participated to this one led by Gabigorski in 2014. I have this Tara Mediterranean from that expedition. And then there was one that explored all the coast of Europe for microplastic in 2019. And the last expedition is not here, but it was around all the coast of Europe, and that was led by the European Molecular Biology Laboratory. And they did also some correlations with the biodiversity of the coast inland. So that's a very ambitious project, which is being analyzed now. Now, just to give you an idea of what kind of plankton I encountered, for example, going around the Arctic in the Bering Sea, these were the major organisms. Of course, the predators were the jellyfish, the ctenophores, and the mollusks. And then there were 3 kinds of abundant crustaceans that were certainly feeding on these diatoms. So this was a very simple ecosystem in terms of the main, very abundant, but 5 or 6 type of main organisms. And then 3 weeks later, we were in the south part in Greenland. And there were 2 kinds of diatoms that was a major protist, and then they were feeding basically 3 types of copepods. So that was the composition of the ecosystem in terms of the main organisms. So in terms of communication, of course, we had constantly I was working with a small team of the Tara Foundation and doing the junction between the team of scientists and the people who were doing the communication. And these were points that we tried to stress, of course, that not only plankton is the basis of the food chain and the ocean productivity, but it is also a major partner or actor for the global geochemical cycles from the carbon pump and so on. and make people understand that phytoplankton produces about 50% of the oxygen and absorbs about 30% of the atmospheric CO2, which is, of course, very important in the context of climate change. And also stress that plankton is really at the base of oil and gas, because it's all this plankton that's settled in the bottom of the ocean that's transformed into our fossil fuels. So these were our main talking points, and we illustrated them with the films and different actions. We also made transversal films to go on platforms that, of course, were much more traveled than Plankton Chronicles, which was mostly used by schools. There are about 300 visits a day, mostly schools. And on TED platforms, this was viewed millions of times, and it tells the story of the food chain using a fish.
How did I get here? Well, it's a stranger story than you might think. I came from a world of drifters, a place few humans have ever seen, the world of plankton. I came from a batch of 1,000,000 eggs, and only a few of us survived. When I became a larva, I moved among other drifters. My fellow plankton came in all sizes, from tiny algae and bacteria to animals longer than a blue whale. I shared my nursery with other embryos and juveniles, from clams and crabs to sea urchins and anemones. We drifting animals are called zooplankton. The most common animals here are copepods and krill. You could search the world over, but you'd never find a place more diverse than my childhood home. A teaspoon of seawater can contain more than 1,000,000 living creatures. It can be a pretty tough existence, though. Trillions are born here, but only a few make it to adulthood. We may be no larger than a pinhead. This crab larva is an arrowworm's worst nightmare. Epic battles between carnivores like these are just one way to get food. But the real powers of this place come from phytoplankton, single-celled life that transforms sunlight and carbon dioxide into edible gold. Phytoplankton are the base for the largest food web in the world. During the night, many animals like me would rise up from the depths to feed on this sun-powered feast. I was part of the largest daily migration of life on Earth. During the day, I'd return to the darkness where I'd join my bizarre companion. Cannibal, like this sea butterfly mollusk that eats its next of kin. And comb jellies that eat cilia like rainbowed eyelashes.
Some of these snare their prey with sticky tentacles, while others just take a bite out of their countenance. And the siphonophores that catch prey with toxic fishing lures. But my favorite would have to be the crustacean Phronema. Its monstrous looks inspired the movie Alien. It can catch tiny bits in its bristles, but prefers larger prey like sauks. With 2 sets of eyes, this female prowls the deeper water. Prey in hand, she performs one of the strangest behaviors in the entire animal kingdom. With body parts from her victims, she delicately assembles a barrel-like home, feeding her young until they can drift off and survive on their own. Best of all, they make the perfect snack for a small fish like me. Here among the plankton, the food web is so tangled and complex, even scientists don't know who eats whom. But I do. At least now you know a bit of my story. There's so much more to me than just a tasty meal.
So I'm not going to go much into the scientific outcome of the Tara Ocean. I would say that it took about 10 years. It took about 5 years for us to publish significant results in science. 5 articles were published that really, gave a meaningful feeling for what the first conclusions were. Basically, that relationships between organisms played a major role, for example, rather than just the environmental parameters. And then there were some very significant results published in Cell, for example, the distribution of the ecosystem from pole to pole, which, as on Earth, is a gradient distribution. But the surprise there is that the viruses turned out to be extremely diverse at the poles. I think the main contribution has been to bring so many new genes into the databases that we see now there is an avalanche of publications using the Tara Ocean data. And that was really the original idea. It took about 10 years for people to realize that there was this treasure and mine it for their protein involved in a pathology of some kind or so on. They can know when that protein appear in this very ancient ecosystem. And the other outcome, of course, was to make plankton better known. And when the 5 articles of Science were released, my photographs were in the front page in Le Monde and New York Times and many media. For the first time, people did, I think, heard about the importance of plankton not being just krill, which is usually what people think. There was a lot of efforts done in communication that was very well organized by a small team at the foundation. At every port of call where we stayed more than 2 or 3 days, sometimes we stayed for a week. If we had, for example, to send samples, or if the next navigation was a long one. We needed to really completely get the food and so on, get the new members, because as a scientist, we stayed about from 2 weeks to 4 weeks on the boat, and the crew stayed about 3 months. And we received a lot of visits on the boat that were very well organized by the team in Paris, months in advance. So we had a lot of communication with the people, with the different places where we stayed. And during the expedition, there were also some films made, some books published. So there was a very big communication program going on. This gave my son Noe and I also the opportunity to, because plankton is so beautiful, it gave us the opportunity to show it in many different places, from Science Museum, of course, but beyond Science Museum, a lot of art museum realized that this would be really of interest to a larger public. And for example, this was a collaboration we had with a Japanese artist, Shiro Takatani, and musician Ryuichi Sakamoto, who passed away last year, actually. And we had this beautiful installation with thousands of sequences of plankton on as a kind of a Zen garden shown in Kyoto and in Paris. We also made a little museum for a beautiful place called Eden Project in UK, and so on. And in recent times, we've had this year several exhibits in Paris, Abu Dhabi, Nice, Montreal, and Noirmoutier, which is a small place on the Atlantic coast, and mostly showing this immersive and interactive installations. This is the immersive installation shown in Paris, where people were surrounded by plankton, and this can be linearized also. And we ran a lot of workshops also with different public, including kids, having the kids observe the plankton, of course, and then draw plankton or take photographs of plankton. It's a very exciting activity. This is a little demonstration of the interactive mandala, where there are about 80 different plankton loops, video loops, and you can choose any one, and then you learn something simple about it, and you can navigate depending on size or depending on whether it's animal or vegetal, some things that kids really like. And it's being showed right now in Paris at the Museum there, which is a museum for kids. Maybe I'm going to skip that. You can look at it on the internet. The most viewed documents that we did is called Manta Ray, and it's on YouTube. And it's basically a video clip for 2 musicians, and it got viewed by millions of people. And it's about extinction. Anthony has done a beautiful poem about extinction. I just want to spend a few minutes on my most recent project, which, as Wad mentioned, this book about cells and the history of life, for which I did drawings. And it basically explains from the origin of life, what we think about viruses, what we think about the most primitive cells and how little by little life became more complex. Here you see several pages of this, including a lot of pages about cell biology, basically. And I had the chance of having exhibits using my drawing in New York and in France. And my current work is a more historical project about how the marine station of Villefranche-sur-Mer and the marine station in general and the biology came together in the 1850s to 1880s at our 30 crucial years, which corresponds to the life of this man called Hermann Voll, who is usually credited with having discovered fertilization. But his life is like extraordinary story, and that's what I'm interested in right now. There are two articles. I think you have the links to this two articles describing basically what happened on the Riviera between 1850 and between 1800 and 1900, and how as biologists we basically followed in the footsteps of these early biologists. So the book is going to be published actually in a week in New York as in English. So if you have the link, I think you might be interested. Okay, so I could answer questions if there are any.
Thank you very much, Christian. Are there questions in the chat? I can start with a question for you, but it's more general, more maybe difficult. It's so for this last book, you did the drawing, the illustrations. We know there are high, advanced techniques of imaging, etc. What the drawing or the art in general brings to the science you are doing? Why this choice? Why illustration in particular? And how this help? If you have to, it's a kind of bridging the art and science, how this helps.
Well, these are the protists, okay? So so everything is color-coded in the book. So the chromosomes are always blue throughout all the drawings. The membranes are white or yellow if it's archaea. The mitochondria are always red. Of course, this you couldn't possibly, and you have a lot more information here than in any modern imaging, and of course, it's based on what we know from modern imaging, but that gives you the ability. Same for viruses. Here are all the viruses that people may have heard of, from herpes to smallpox to Ebola and COVID, or the giant viruses. This would be very difficult to do in just imaging coming from the machines that we have or the computers. This is just complementary as a way to extract interesting information obtained by these devices into something that anybody can understand.
Thank you. Any questions from the chat or from the audience? Oscar?
Hello, can you hear me?
Yes.
Yes. I don't have a question. I just have a comment. I wanted to thank you because, well, you gave us a nice reminder of something which I think is very important in science, which is beauty, and the search for beauty, which it's easy to forget as we work every day, on our little bit of science, and also about the power that this beauty has to inspire and to transmit um, our science to the general public. So I just wanted to say that I think what you are doing is really really important, not only scientifically but um, also in terms of communication and to remind us of these questions.
Yeah, there
So
is
thank
no
you.
question that there is a very critical attitude towards science that's developing, which is very worrisome. And one way to capture the attention of people who would not even spend a minute thinking about scientific thing would be through this aesthetic aspect, artistic aspect. I mean, it basically touches a population that has kind of given up on science because of school or whatever complotic. I mean, so it's important that we can reach a larger part of the population, not just the part that is willing to listen to science. Yeah.
Completely agree. Thank you.
And also as to attract young people, showing them that there is beauty in science. It's very important.
Agreed. Any other questions or comments from the audience? Hans, Peter?
Yes, thank you very much for this fantastic presentation. I have a more general question as well. How much did you include school classes and other public persons to actually sell the topic a bit? Because what I really like from your images is that they give a new perspective on how to look at plankton in general, and also explain complex processes or food web dynamics and other issues, complex issues in a rather simple and understandable way?
Well, I know that the Plankton Chronicles is used a lot by school, and we designed the films to be very short, 3 to 5 minutes. So it's probably a time of attention that fits with the contemporary level of attention. So the schools use it quite a bit because they have in 5 minutes they basically have anything that can be said really important about an organism.
And did you get some response from the public? And I'm also asking, was there also some funding initiative due to your films and presentations and
Well,
public art?
yeah, I've given lots of talks and to different audiences. And also on Tara, there was a school program that was coupled with the expedition. And sometimes we even had a connection, a satellite connection to the schools. So this was used actually as a support for lots of teachers to talk about oceans, life in oceans, biodiversity. And the foundation actually published a little booklet and also some pedagogical materials that were quite well used, and with time it's been almost institutionalized. So there is a big program coupled with the expeditions in terms of having schools participate. And that's
Yeah, I think that's really great
yeah,
because
yeah,
it forms the future scientists, huh?
yeah. Of course, expedition, there is the aspect of the adventure that's very attractive. We particularly for sciences that are laboratory sciences where people stay inside and in front of computers. I think a lot of people perceive that this not as attractive as it could sound. And this puts back some adventure and some, and it's true that we were very courageous and in some way crazy because we didn't get funded really. But that made it also easy because we didn't have any, big pressure. It took us 5 years to publish the first things. But nobody was, putting us under pressure. So this is science can happen outside of the tight project that most science is tied to. But it's certainly an exception.
Yeah, I like to hear this because I think we need to have some freedom or sometimes more freedom in science aside of administration and other obligations to actually do the stuff we want to do, right?
Yeah.
That's
What was
fantastic.
interesting, I must say that what was interesting is that at least two institutions, life science in the CNRS, the French research institution, and the European Molecular Biology Laboratory, they did sense our enthusiasm. And then they found a little bit of money here and there to help us when needed. And they didn't put any, they didn't try to stop us because some other departments in the CNRS were very adversary because, of course, we were co-opting, for example, some engineers to go on the expedition, and they didn't quite like that because they thought they should stay within their labs inside and not go into some things that they thought was, too adventurous, maybe. Yeah. And also the oceanographic community was very dubious that we could do anything good on a small sailboat like that. But we had a really good professional crew, and we had some good engineers with us that knew how to adapt the boat to sample properly. So, as I said, there was not a single place where we couldn't sample.
Perfect. Thanks.
Well, thank you very much. Thank you very much, Christian, for joining us today. Thank you, everyone. And I will say Happy New Year from now, and I'll see you next year.
Okay.
Thank you very much.
Bye.
Bye.